Now starting with the parasympathetic system. Okay, now the important neurotransmitter of parasympathetic nervous system is acetylcholine. Okay, now here there is one request, you should know the structure of acetylcholine. I don't have more space to write it on the board properly. Okay, now acetylcholine is the important neurotransmitter. आता neurotransmitter or the neurons, ठीक है, ब्रेन मध्ये जेव्हा नर्व ऍक्शन पोटेन्शियल किंवा नर्व्स इम्पल्सेस ट्रॅव्हल करतात, हे खूप हाय स्पीडने प्रोसेस होते. ओके, अँड इवन व्हेन यू हॅव टू रिलीज द न्यूरोट्रान्समीटर, दॅट इज ऑल्सो व्हेरी फास्ट प्रोसेस. ओके, आता या केस मध्ये तुम्हाला न्यूरोट्रान्समीटर हे प्री सिनॅप्स मध्ये, ठीक आहे, सिनॅप्सच्या आधीच्या न्यूरोनल स्ट्रक्चर मध्ये हे ऑलरेडी प्रेझेंट पाहिजे. ठीक आहे, इट शुड बी स्टोर्ड देयर. नाउ means these neurons should be able to synthesize themselves. ठीक आहे, म्हणजे त्यांना स्वतःला त्या न्यूरोट्रान्समीटरचं सिंथेसिस करता आलं पाहिजे. ओके, सो द सिंथेसिस ऑफ मोस्ट ऑफ द ऑल न्यूरोट्रान्समीटर इज टेक्स प्लेस इन दोज न्यूरॉन themselves. ठीक आहे, नाउ हियर वी आर गोइंग टू डिस्कस द बायोसिंथेसिस ऑफ एसिटिलकोलिन व्हिच टेक्स प्लेस इन द कोलिनर्जिक नर्व इटसेल्फ. ठीक आहे, आता इथे तीन रेड कलर मध्ये इंपॉर्टंट हेडिंग दिलेले आहेत. फर्स्ट हेडिंग इज एसिटिल कोए सिंथेसिस, सेकंड हेडिंग कोलिन अपटेक फ्रॉम आउटसाइड ऑफ द न्यूरॉन, थर्ड हेडिंग द एसिटायलेशन ऑफ कोलिन. ओके, आता या केस मध्ये दिस फर्स्ट अँड सेकंड याला आपण क्लिअरली फर्स्ट अँड सेकंड ने म्हणू शकतो. दिस टू स्टेप्स कुड बी ऑकरिंग साइड बाय साइड. ओके, नाउ स्टार्टिंग एसिटिल कोए सिंथेसिस इनसाइड द न्यूरॉन. ठीक आहे, न्यूरॉनच्या स्ट्रक्चर आतमध्ये एटीपी, एसिटेट अँड कोएन्झाइम हे कंबाईन होतात. दिस फॉर्म्स द एसिटिल कोए, सॉरी, this combines to form the acetyl CoA. Then side by side what is happening? Choline uptake from the outside. Here sodium choline cotransporter is present on the membrane which uptakes the choline molecule. Now step 1 मध्ये फॉर्म झालेला acetyl CoA and this 1b मध्ये फॉर्म झालेला choline. ठीक आहे, this acetyl CoA and choline will combine and it is clearly the second step of biosynthesis of acetylcholine. Okay, this step is known as the acetylation of choline molecule. We have acetyl group present here and we have choline, means ह्या uptake केलेल्या choline molecule चं काय करणार आहे आपण? Acetylation, and this will lead to the generation of acetylcholine. Okay, so it is very much easier to understand. ठीक आहे, खूप ब्रीफ मध्ये आपण पुन्हा रिवाइज करूया. एसिटिल कोए सिंथेसिस फ्रॉम एटीपी, एसिटेट अँड कोएन्झाइम. इथे काय तयार होणार आहे? एसिटिल कोए. देन एसिटिल कोए तयार झाला, हे सगळं कुठे झालं? न्यूरॉनच्या आतमध्ये. ऍट द सेम टाइम कोलिन विल बी अपटेकन फ्रॉम द आउटसाइड ऑफ न्यूरोनल स्ट्रक्चर. दिस कोलिन कम्स फ्रॉम द outside the neuron. Now acetyl CoA and choline combination. This step is known as acetylation of choline molecule and it leads to the formation of acetylcholine. Okay, in this way your neurotransmitter of parasympathetic system is generated. This event took place inside the neurons. Now neurons मध्ये पण acetyl CoA ला एक फ्री फॉर्म मध्ये रिलीज नाही केलं जात. These are transmitted inside the vesicles. Okay, in these vesicles acetylcholine is stored in the ionic solution. Okay, now you are having that structure of acetylcholine. It have that positively charged nitrogen. ठीक है, so it makes that necessary to store it in the ionic solution. Okay, so these vesicles are almost of 40 nanometer in diameter. 40 nanometers only. Okay, 40 nanometers. ठीक है, so these are stored here and when the action potential allows, this acetylcholine is released outside the neuron. How? This vesicle fuse with the membranes of neurons. Okay, and then releases this outside. Okay, now here this complete biosynthesis is affected by certain molecules or the chemicals. Okay, now this step, the choline uptake step, is affected by hemicolinium. Hemicolinium blocks the choline uptake. ठीक है, इथे एक पार्ट झाला. Second part, when the acetylcholine is synthesized, Vesami calls blocks the transport of acetylcholine into the vesicles. Okay, it's a second step. Then second toxin or the poison which is acting, then acetylcholine release by exocytosis, or this release process is affected by black widow toxin. Okay, and here this release is affected, means it avoids the release or it inhibits the release of acetylcholine, and then one toxin is present, that is the botulinum toxin, okay, which causes the massive release of acetylcholine. Okay, means त्याचा इफेक्ट म्हणजे त्याचा कॉन्सिक्वेन्स काय होणार की न्यूरॉन्स will suffer from the complete or almost all loss of the acetylcholine. Now you have studied the biosynthesis of acetylcholine. Okay, 3 easy steps मध्ये होतं. Now just revise those one. ठीक है, it is not written on the board. Which one? The first one was synthesis of acetyl-CoA, second one was the choline uptake, and the third one was synthesis of acetylcholine molecule. What happens after the synthesis of acetylcholine? After the synthesis of acetylcholine, it is transported into the vesicles. Okay, and depending on the need, it is released. Okay, inside the vesicles, it is stored in the ionic solutions. Okay, so हे सगळं आपण डिस्कस केलं. Now how it works? Here the action potential reaches to this cholinergic neuron. Okay, now because of this action potential, the calcium levels inside these neurons are increased. Okay, as a result of increased calcium levels, these vesicles, which were the storehouse of acetylcholine, starts to migrate toward the neuronal membrane. Okay, toward the synapse. Okay, the neuronal membrane which is facing toward the synapse. How these vesicles will know to travel toward this direction, or they have to travel toward this opposite direction? Okay, for that there are certain proteins inside the neurons. Okay, which helps to fuse, which helps to travel this vesicle in the proper direction. This helps to attach with the neuronal membrane, which help to release their secretion outside the neuron, and which helps to retrieve them. Okay, blank vesicles ला पुन्हा collect करणार. How it will happen? Now first of all, the calcium levels are increased. Okay, immediately the vesicle trafficking proteins helps to travel these vesicles in the direction of this the membrane which is facing toward the synapse. Now once these vesicles are reached to the membrane which is facing here, they are attached with this neuronal membrane. Okay, this attachment of vesicle with the neuronal membrane is known as the docking, which is nothing but used to stop the boats in the sea, at the seashore. Okay, that is from there the docking comes. Now once these are docked on the neuronal membrane, you need something to firmly attach. The vesicle membrane should be attached with the neuronal membrane, that is known as the priming. So there are different proteins of docking, for priming and then fusion. Okay, now here calcium is necessary to fuse the vesicular membrane with the neuronal membrane. At the same time, this calcium is necessary to fluidize this vesicular membrane. Now what will happen? Vesicular membrane ला fluidize करना means somewhere that pores create करना, means fluidity create करना, more transparency in that vesicular membrane. So acetylcholine can come out of the vesicles. ठीक है? Now here after fusion, membrane is fluidized. ठीक है? Once membrane is fluidized, that acetylcholine is released outside the vesicles. ठीक है? Means outside the neuron, and this process is known as the exocytosis. ठीक है? Now here, apart from these neuronal proteins, on the surface of vesicle, the synaptogramins are present. These are the proteins which help in the exocytosis process. ठीक है? Now once the acetylcholine is released outside the neuronal membrane, it diffuses in the synapse. ठीक है? Synapse is nothing but what? The gap between these two neurons. Okay, here it could be organ, here it could be the neuron. So they travel across the synapse. Now at this end, okay, now at this receiver end, there are the receptors of cholinergic transmission. ठीक है? Now this acetylcholine molecule acts on the receptor, and depending on the nature of receptor, the post-synaptic potential is reduced. Now the receptors of acetylcholine, okay, for the cholinergic system. Now there are two major categories of the receptor, okay, which are divided on the basis of agents which active them, okay. So muscarine and nicotine are two different agents which activate the cholinergic receptor, okay. Now those receptors which are activated by muscarine are known as muscarinic receptor, okay, and those receptors which are activated by nicotine are termed as the nicotinic receptor. So these two are the major categories of cholinergic receptors. Okay. Now the first category was the muscarinic receptor. In this category there are total 5 types of receptor are found till now, okay, which are M1, M2, M3, 4. Now you have studied the biosynthesis of acetylcholine, okay, 3 easy steps मध्ये होता. Now just revise those one, okay, it is not written on the board. Which one? The first one was synthesis of acetyl-CoA, second one was the choline uptake, and the third one was synthesis of acetylcholine molecule. What happens after the synthesis of acetylcholine? After the synthesis of acetylcholine, it is transported into the vesicles, okay, and depending on the need, it is released, okay. Inside the vesicles, it is stored in the ionic solutions, okay. So हे सगळं आपण डिस्कस केलं. Now how it works, how parasympathetic system works, ठीक आहे, म्हणजे इफेक्ट कसा प्रोड्यूस होतो और केव्हा प्रोड्यूस होतो, ठीक आहे. Now suppose this is the cholinergic neuron, okay. Now in this cholinergic neuron, what happens here? The action potential reaches to this cholinergic neuron, okay. Now because of this action potential, the calcium levels inside these neurons are increased, okay. As a result of increased calcium levels, these vesicles, which were the storehouse of acetylcholine, starts to migrate toward the neuronal membrane, okay, toward the synapse, okay, the neuronal membrane which is facing toward the synapse. How these vesicles will know to travel toward this direction, or they have to travel toward this opposite direction, okay. For that there are certain proteins inside the neurons, okay, which helps to fuse, which helps to travel this vesicle in the proper direction. This helps to attach with the neuronal membrane, which help to release their secretion outside the neuron, and which helps to retrieve them, okay. Blank vesicles ला पुन्हा collect करना. How it will happen? Now first of all, the calcium levels are increased, okay. Immediately the vesicle trafficking proteins helps to travel these vesicles in the direction of this the membrane which is facing toward the synapse. Now once these vesicles are reached to the membrane which is facing here, they are attached with this neuronal membrane, okay. This attachment of vesicle with the neuronal membrane is known as the docking. So which is nothing but used to stop the boats in the sea, at the seashore, okay. That is from there the docking comes. Now once these are docked on the neuronal membrane, you need something to firmly attach. The vesicle membrane should be attached with the neuronal membrane, that is known as the priming. So there are different proteins of docking for priming and then fusion, okay. Now here calcium is necessary to fuse the vesicular membrane with the neuronal membrane. At the same time, this calcium is necessary to fluidize this vesicular membrane. Now what will happen? Vesicular membrane ला fluidize करना means somewhere that pores create करना, means fluidity create करना, more transparency in that vesicular membrane. So acetylcholine can come out of the vesicles, okay. Now here after fusion, membrane is fluidized, okay. Once membrane is fluidized, that acetylcholine is released outside the vesicles, okay, means outside the neuron, and this process is known as the exocytosis, okay. Now here, apart from these neuronal proteins, on the surface of vesicle, the synaptogramins are present. These are the proteins which help in the exocytosis process, okay. Now, once the acetylcholine is released outside the neuronal membrane, it diffuses in the synapse. Okay, synapse is nothing but what? The gap between these two neurons. Okay, here it could be organ, here it could be the neuron. So they travel across the synapse. Now at this end, okay, now at this receiver end, there are the receptors of cholinergic transmission. Okay, now this acetylcholine molecule acts on the receptor, and depending on the nature of receptor, the post-synaptic potential is reduced. Till now, you have studied how acetylcholine or parasympathetic system works. Okay, so you know that the major neurotransmitter for parasympathetic system is acetylcholine. I hope you have the structure with you right now. Okay, now after that, consider the presynaptic structure, synapse, and postsynaptic structure. Okay, now when acetylcholine is secreted, it diffuses toward the postsynaptic structure. Now here, to show some effect, it will need a presence of receptor. Okay, acetylcholine have the two different types of receptor, muscarinic and nicotinic. Now how this classification has been done? Now the basis of classification is the agent which is activating that receptors. Okay, now in the initial phase of development or the research work, okay, it was found certain group of receptors of parasympathetic system was activated by nicotine only. Okay, while the other group was activated by muscarinic only. So the two groups of receptors of parasympathetic system has found. Accordingly, they were named as muscarinic receptors and nicotinic receptors. Okay, but this is not enough. Okay, now if agent is activating, the receptor is activated, so call it as the muscarinic or nicotinic. There was an underlying difference. Okay, and which was that difference? Here it is written. Muscarinic receptors are typically the G protein coupled receptors, while the nicotinic receptors are ligand gated receptors. To activate this particular group of receptor, you will need some structural features which were fulfilled by muscarinic, and same application is for this category. We have two broad categories, the muscarinic one and the nicotinic one. Now the muscarinic receptors are further classified into 5 subtypes: M1, M2, M3, M4 and 5. Okay, so here it is written. At the same time, nicotinics are classified as NM and NM. Okay, here numbering is given and here the location of the tissue is given. Okay, so discussing this section first. Muscarinic receptor M1, M3, M5 are GQ coupled receptor. Here the word is written. Okay, GQ, GQ and GQ. While M2 and M4, okay, the separate color is used, are GI protein coupled receptor. Now what happens when it is GQ coupled receptor? It works by IP3 diacylglycerol pathway, means inositol triphosphate and diacylglycerol pathway. As a result, the intracellular calcium increases. Now we have studied, okay, postsynaptic potential part. There is excitatory postsynaptic potential and inhibitory postsynaptic potential. Recall that slide. Okay. Now when the intracellular sodium or calcium increases, the activation occurs. Okay, so here what will happen with M1, M3 and M5? Because of this pathway, the postsynaptic structure will be activated. Okay, that's why M1, 3 and 5 are called as excitatory receptors, while M2 and M4, here the G protein coupled is GI. When the GI protein is activated, the intracellular calcium decreases, means the potassium outflux occurs. Okay, here the intracellular cyclic AMP decreases. Now this arrow might be confusing, so let me erase them. Okay, potassium outflux occurs. At the same time, the cellular content of cyclic AMP means intracellular cyclic AMP decreases. As a result, the postsynaptic structure gets inactivated. That's why M2 and M4 are inhibitory receptors. Now, very short mnemonic to memorize this. 135 are excitatory receptor, while 2 and 4 are inhibitory muscarinic receptor. Now, few more words about it. M1 receptor typically forms in the central nervous system. That's why it is also called as the neuronal receptor. Then M2 typically found on the heart cells. That's why it is called as the cardiac receptor. Okay. At the same time, M3, which is found on smooth muscles as well as glands. Okay. Then these three receptors are primarily found. Okay. While 4 and 5 are lesser. They are physiologically not significant. Okay. That's why while studying this chapter, our focus will be on 1, 2 and 3rd muscarinic receptor only. I hope you got this description. Now moving to the next section, that is the nicotinic receptor. It is ligand gated receptor. Okay. So here there are two types on the basis of location of receptor. NM refers to those nicotinic receptors which are present on skeletal muscles. And N receptor refers to those receptors which are present on the autonomic ganglia. Autonomic system belongs to the peripheral nervous system. When we are saying that ganglia, it refers to cluster or group of PNS neurons cell bodies. Cell body refers to the structural part of neuron. So here group of cell body exists as a ganglia. Now this ganglia or the group of cell body, it could belong to the parasympathetic system or sympathetic system. Now if consider this ganglia belongs to the sympathetic system, then on activation which system will be further work? That will be the sympathetic. And if the ganglia belongs to parasympathetic, then which system will work further? That will be the parasympathetic system. Now when we say the sympathetic outflow from ganglia, means sympathetic system will work further, and when we say the parasympathetic outflow, the parasympathetic system will work further. Okay. Now what happens when this NN receptor activated, one or this one, okay, will be activated further. When this NM receptor is activated, skeletal muscles undergo contraction. Okay. This you have to understand properly, especially this part. Now discussing the M1 receptor first. It is very much easier to remember. I know every one of you able to recall all these things once you have seen on the board. Okay. Now let's explore this part. Autonomic ganglia. You have also read this word in NN section, the nicotinic neural receptor. See, the location of those neural ganglia are away the central nervous system, but these autonomic ganglia are present in the regions of central nervous system. Okay, so when this M1 receptor over these autonomic ganglia are activated, it causes depolarization. Okay, now depending upon the functional role, the effect will be observed. The second one, CNS, cognitive work. What is meant by cognitive work? Learning, memorizing or thinking. Three easy words I have given to you. Learning, memorizing and thinking. Okay, all this determines the cognition. So what happens when the acetylcholine in the brain is increased, okay, or it is somewhere released, that is responsible for the cognitive work. When the M1 receptors over the gastric cells are activated, they causes the release of histamine. Okay. Now, thinking and histamine release. How you are going to connect it? When you overthink something, okay, when thinking is much more, okay, unnecessary thinking is there. At that time, what happens? Acetylcholine dominates. Okay, and this somewhere linked with this. Okay, and gastric cells on M1 receptor activation leads to the release of histamine, and this histamine causes the acidity in stomach or ulcers. Okay, so in this way your overthinking is also related with the generation of ulcers in the gastric system. Okay, so these three are the important locations of M1 receptor in brain. That's why it is also called, sorry, two only. This third one is in the gastric system. So because of these two, where the M1 receptor is chiefly found, it is also known as the neural receptor. Now we studied the location and the functional effect. These are mentioned the selective agonists, which includes xanomeline, cevimeline, MCN343A, then pyrazine and telangiecine. Okay. So now moving to the next part, that is the M2 receptors. Here the locations are given: SA node, AV node, and the fibers, the atrial fibers or ventricular fibers. All these locations represent the heart. Hence the M2 receptor, because the M2 receptor chiefly or only found on the heart cells, that's why it is also known as the cardiac receptors. Okay. Now what are the effects of M2 receptor activation on heart? Let's see. SA node, okay, from where the impulses for cardiac contraction generates. Okay. Now what happens on M2 receptor activation? These SA nodal cells will undergo hyperpolarization. Now these terms, depolarization refers to excitation, while repolarization or hyperpolarization refers to the inactivation. So here SA nodal cells will somewhere undergo inactivation. What is the role of SA nodal cells in the heart? The impulse generation for cardiac contraction. And since it is now somewhere inactivated, what will happen? The rate of impulse generation will be decreased, or the intensity of impulse generations will be decreased. Now one note I have to present you over this part. Okay, or when you are studying that classification of receptor of this particular receptor M1, M2, M3 revised reading in the book. ठीक है, तो त्यावेळेस तुम्हाला एक केअर घ्यायची. Do not undergo directly into the deep pharmacology starting with this. ठीक है, when you are reading M1, M2, M3 receptor, okay, initially directly deeper pharmacology चे correlations इथे apply नका करू. ठीक है, it will surely create you confusion. ठीक है, once you understood the complete ANS, then come back to this part. Okay, so till now just start. So SA nodal cells hyperpolarization means what? It will not be able to generate an impulse. As a result, the rate of impulse generation will be decreased, as well as the intensity of impulse generation is also reduced. Second, AV node. After the SA node, impulse propagates toward the AV node, okay, and then bundle of his and then Purkinje fiber. Now here both AV node and the Purkinje fiber. Here what happens? The refractory period is increased. Means refractory period means what? These cells will not respond to the next impulse in this time. Okay. Now this time to respond to the new impulse is now increased. Even if SA node is sending you 60 impulse to contract. Okay. But because of this increased refractory period, all these impulses will not be propagated in a fixed time. The time will be increased. So because of this, the heart rate is reduced. Okay. Then atrial and ventricular fibers. Here the contractility of these fibers is decreased. So concluding all these effects, the ability of heart to the work is decreased. Why? Because of M2 receptor. And this is known as the parasympathetic influence on heart. Okay. Now the agonist part. Okay. There are two selective agonists, that is the berberine and methaqualone. Okay. While the antagonists are methotrexate and tryptamine. Now the M3 receptors. See, the location of M3 receptor is diverse. You cannot classify that it only belongs to the muscles or it only belongs to the glands. Okay. Now the smooth muscles. Okay. When the M3 receptor located over the smooth muscles, whether it could be the bronchial smooth muscles or vascular smooth muscles, the muscles will undergo contraction. Okay. When the M3 receptor located on circular muscles of iris, you might be able to see that image with this. The circular muscles of iris constrict. As a result, meiosis occurs. What is meant by meiosis? Meiosis refers to the constriction of pupils. These two belongs to the eye. Okay. Ciliary muscles also constrict. Because of constriction of ciliary muscles, the spasm of accommodation occurs. Now, what is the meaning of spasm of accommodation? Means you are not able to rotate the eyes. Okay. Then the vascular endothelial cells. Vascular refers to blood vessels. Now endothelial layer, the innermost layer of blood vessels. Here what happens when the M3 receptor located on that innermost layer, okay, blood vessels चा जो innermost layer असतो तिथे M3 receptor आहे, and when that M3 receptor is activated, that causes release of nitric oxide. This nitric oxide is also known as endothelial dependent relaxing factor. Its short form is EDRF, that is endothelial dependent relaxing factor. Now it is synthesized from arginine, okay, the amino acid. Now the calcium calmodulin pathway is used for the synthesis of NO. Now what happens because of this nitric oxide release? Now answer is here. Endothelial dependent relaxing factor means blood vessels are relaxed, means vasodilation occurs. So it is the effect of M3 receptor activation. Moving to the next part, that is the glands. Now you know there are lacrimal glands, salivary glands, sweat glands, then biosecretion glands and many more. Okay. Now what happens? There are certain glands which receive the parasympathetic innervation. Okay. What word I have used? Parasympathetic innervation. Means जिथे जिथे ज्या ग्लँड्स वरती पॅरासिम्पॅथेटिक इन्फ्लुएन्स एक्झिस्ट करतो, ठीक आहे, wherever the parasympathetic innervation is present, now all those glands only on M3 receptor activation causes the secretion. Okay, if that is the sweat gland, that will cause the sweating. If that is the lacrimal gland, that will cause the secretion of fluid over there. If that is the salivary gland, will cause the secretion of saliva. Okay, so these are the effects of M3 receptor activation. Relatively selective agonist. Now here wording is changed. I have used the word relatively selective, means these drugs have more preference toward the M3 receptor as compared to M2 or M1 receptor. Such phenomena is known as the relatively selective agonism. Now bethanechol, carbachol and oxytrimolene, these are the agents, while antagonists are solifenacin and darifenacin. ठीक है? So here we have completed M1, M2 and M3 receptor study.